Good afternoon to all of you. Myself, Dr. Sandeep Kotwal. I am Senior Assistant Professor of Botany at Government Degree College, Bhadrava. Today, the topic of my presentation and discussion is seed formation, structure of monocot and dicot seeds. So, let us start. What are seeds? Seeds are the end products of sexual reproduction which are formed in both gymnosperms and angiosperms. That is why the gymnosperms and angiosperms are known as the seed-bearing plants or the spermatophytes or the phanerogames. Whereas those which reproduces by the spores, they are known as the cryptogams and they are the algae, bryophytes and pteridophytes. The seeds are very essential for sexual reproduction as they are the embryonic plants. In gymnosperms, the seeds are naked because they have no flowers or fruits, whereas in angiosperms, they are formed from the matured ovules that are enclosed within the fruit, which is developed from the ripened ovary after fertilization. Angiospermic seeds are formed after double fertilization and they consist of three genetically distinct constituents: the embryo, which is formed from the zygote, the triploid endosperm, and the seed coat, which is derived from the maternal tissue of the ovule. A typical seed contains a seed coat, cotyledons, endosperm, and a single embryo. The seeds are generally the matured ovules. Dear friends, we are very much familiar with the different types of seeds. So this slide shows the different shapes, sizes, and types of the seeds. The immature ovules consist of a number of components, and they are the funicle or the seed stalk, which attaches the ovule to the placenta and hence ovary or fruit wall at the pericarp. The nucellus, the remnant of the megasporangium and main region of the ovule where the megagametophyte develops. The micropyle, a small pore or opening in the apex of the integument of the ovule where the pollen tube usually enters during the process of fertilization. And the clausa, the base of the ovule opposite the micropyle where integument and nucellus are joined together. The final shape of the seeds is often affected by the shapes of the ovule. Plants generally produces 6 shapes of ovules, and they are the orthotropous or the atropous ovules, the anatropous ovules, the hemianatropous ovules, compylotropous ovules, amphitropous ovules, and the circanotropous ovules. This slide shows the different types of ovules. The figure A is the orthotropous ovule and it is generally present in the gymnosperms. In this case, the ovules are straights in which the clausa, the funicle, and the micropyle, they all are aligned up in a long row producing an uncurved seed after fertilization. Figure B is the anatropous ovule. This is the most common ovule which is present in the angiosperms, and in this case it has a curved shape in which the ovules become completely inverted at 180 degree during development so that the micropyle lies close to the hilum. The hilum is a scar that marks the point where the seed was attached to the fruit wall by the funicle. Figure C, it shows the hemianatropous ovule in which the body of the ovule bent at a right angle, that is at 90 degree in relation to the funicle, so it looks like the ovule is lying on its side. Next is the compylotropous ovules. In this type, the body is bent and the alignment between the clausa and micropyle is lost. The embryo sac is only slightly curved, often giving the seed a tight C shape or kidney shape. Figure is of the amphitropous ovules. In this case, the body of the ovule is very much curved so that the embryo sac and the ovule itself take the shape of a horseshoe. Next is the circanotropous ovules, in which the funicle is especially long so that it creates nearly full circle, that is 360 degree around the ovule whose micropyle is ultimately pointing upwards. Now friends, move to the seed formation and development of a seed. Ovules are the precursors to seeds and as such are critical to plant propagation and food production. A seed is formed when fertilized ovule divides by mitosis. It stores food and has the potential to develop into a new plant under optimal conditions. In angiosperms, the process of seed development begins with the double fertilization, which involves the fusion of two male gametes with the egg cell and the central cell to form the primary endosperm and the zygote. This slide shows the double fertilization, and this is a depiction of a anatropous ovule. Pollen grains are transferred to stigma by various pollinating agents such as water, wind, butterflies, insects, animals, birds, etc. The pollen grains which falls on the stigma, then they forms the pollen tube. This pollen tube reaches at the micropylar end from the style region of the carpel, and then at the micropylar end it bursts open and it releases the two male gametes which are present in it. One male gamete, it fuses with the egg cell and it forms the zygote, whereas the other male gamete, it fuses with the primary endosperms nuclei, and then it forms the endosperm, which is a triploid. So this is the double fertilization. Right after fertilization, the zygote is mostly inactive, but the primary endosperm divides rapidly to form the endosperm tissue. This tissue becomes the food for the young plant, which will be consumed until the roots have developed after germination. The so formed zygote divides and develops into an embryo. Following the fertilization, every pod of the flower sheds off except ovary. The ovary of the flower develops into the fruit, while ovule develops into seeds. The formation of seed completes the process of reproduction in plants. Within the seed, the growing embryo develops and matures. In the majority of flowering plants, the zygote first region is transversely oriented in regards to the long axis, and this establishes the polarity of the embryo. The callosal pole becomes the main area of growth of the embryo, while the micropylar pole produces the stalk-like suspensor that attaches to the micropyle. The suspensor absorbs and manufactures nutrients from the endosperm that are used during the embryo growth. The main components of the embryo are the cotyledons or the seed leaves, which are attached to the embryonic axis. There may be one or two cotyledons. The cotyledons are also the source of nutrients in the non-endospermic dicotyledons, in which case they replace the endosperm and are thick and leathery. In endospermic seeds, the cotyledons are thin and papery. Dicotyledons have the point of attachment opposite one another on the axis. Then there is the epicotyle, the embryonic axis above the point of attachment of the cotyledons. The plumule, the tip of the epicotyle, and has a feathery appearance due to the presence of young leaf primordia at the apex and will become the shoot upon germination. The hypocotyle, the embryonic axis below the point of attachment of the cotyledon, connecting the epicotyle and the radicle, being the stem root transition zone, and the radicle. The basal tip of the hypocotyle grows into the primary root. Monocotyledonous plants have two additional structures in the form of sheaths. The plumule is covered with the coleoptile that forms the first leaf, while the radicle is covered with the coleorhiza that connects to the primary root and adventitious roots from the sides. So now move towards the structure of the seeds. Based on the number of cotyledons, the seeds are divided into two types: monocotyledonous and dicotyledonous. Structure of monocot seed. The embryos of monocot seeds have only one large cotyledon, and that is called the scutellum. This scutellum is generally shield-shaped and is located laterally towards the side of the embryo axis. In monocot seeds, the endosperm is covered from a proteinaceous layer called aileron layer. The embryo axis of monocots consists of shoot tip, plumule enclosed in a sheath called coleoptile, and a root tip radicle which is enclosed in a coleorhiza. Most of the monocot seeds have thick swollen endosperms for nourishment. During the embryo development, the endosperm is not completely consumed and it is the nourishing tissue in seeds. However, some monocots like orchids show the exceptions. The examples of monocot seeds are the rice, wheat, bamboo, palm, coconut, ginger, onion, garlic, lilies, tulips, etc. In monocot seeds, the test and tegmen of the seeds are fused. As the seed germinates, the primary root emerges, protected by the root tip covering the coleorhiza. Next, the primary shoot emerges, protected by the coleoptile, the covering of the shoot tip. Upon exposure to light, that is, when the plumule has exited the cell and the protective coleoptile is no longer needed, elongation of the coleoptile ceases and the leaves expand and unfold. As the other end of the embryonic axis, the primary root dies, while other adventitious roots, that is, the roots that do not arise from the usual place, they emerge from the base of the stem. This gives the monocot a fibrous root system. So this figure depicts the germination in monocot seeds. Now let's move to the structure of dicot seeds. In dicots, the embryo has an embryonic axis and two cotyledons. The appearance of cotyledons is swollen as they preserve the food for developing seedlings. There are two ends for the embryonic axis. The other end, that is the one end which forms the shoot tip, is called plumule, and the portion of the lower end which forms the root tip at the lower end is called the radicle. The whole content is covered within a protective cover called the seed coat. The seed coat is made up of an outer layer called testa and an inner layer called as tegmen. Moreover, the seed is attached to the fruit through a structure called hilum. The examples are the peas, the almonds, the cashew, the apples, the plums, peaches, bitter gourds, castor seeds, mango seeds, neem seeds, papaya seeds, etc. The dicotyledonous seeds, they are of two different types, also based on the presence or absence of the endosperm. In endospermic dicots, the food reserves are stored in the endosperms, and that's why they are also known as the albuminous seeds. During germination, the two cotyledons therefore act as absorptive organs to take up the enzymatically released food reserves, much like in the monocots. The examples are the tobacco, tomato, the castor, the pepper seeds, etc. All gymnosperm seeds are also albuminous, that is endospermic seeds. In non-endospermic dicots, the triploid endosperm develops normally following double fertilization, but the endosperm food reserves are quickly remobilized and moved into the developing cotyledons for storage. That is why they are also known as ex-albuminous seeds. The two hops of peanut seed, that is Arachis hypogaea, the split seeds, that is Pisum stivum, the walnut seeds, the bean seeds, they all are examples of the ex-albuminous seeds. So this is the figure which shows the structure of the dicot and monocot seeds. Now move to the germination of dicot seeds. Upon germination in dicot seeds, the epicotyle is shaped like a hook with the plumule pointing downwards. This shape is called the plumule hook, and it persists as long as germination proceeds in the dock. The figure B shows the germination in dicot seeds. Therefore, as the epicotyle pushes through the tough and abrasive soil, the plumule is protected from damage. Upon exposure to light, the hypocotyle hook straightened out, the young foliage leaves face the sun and expand, and the epicotyle continues to elongate. During this time, the radicle also grows and produces the primary root. As it grows downward to form the taproot, lateral roots branch off to all sides, producing the typical dicot taproot. So this figure depicts the both germination in the monocot seeds as well as in the dicot seeds. With this, I am very thankful to all of you for the patience hearing. There are certain seeds in which the seed germination occurs while the seeds are present inside the fruits, and these seeds are known as viviparous seeds, and the best example is that of the mangrove seeds. Dear friends, the smallest and lightest seeds are of the oricus seeds, whereas the largest and heaviest seeds are of Lodicea seeds. So thank you very much. I hope you all understand about the seed structure and seed germination. Thank you very much.